So a lot of it is a little bit more tailored towards nematodes, but I tried to throw in a few other topics that might be of interest if you're planting more of an almond orchard. So the first thing I'm going to go over is just like generalized idea of like what actually can cause problems when you're planting an orchard, and potentially some things you can consider based on what it is. But if you get anything, identifying a potential problem and how it might interact with characteristics for that orchard is really important in trying to figure out what mitigation measures can be done, because there might be some that are better to do prior to planting, and there might be options that can be done after planting, because there is no silver bullet, no matter how much we might want to find it. So some things are better at controlling things than others. So knowing what you're going to be dealing with is very helpful for a very long-term investment. I generally like to classify it down to aggressive pathogens and pests, physical and chemical factors or abiotic factors. And while plant-parasitic nematodes is a pest, I tend to like pull it out on its own, especially when talking about planting and replanting almond orchards. So one tool I find very helpful is this combination. So Google Earth is a free program. You can download it on computer. Why I like it is because you can go back and forth and see what that plot of land is doing all the way back for several years. And seeing that progression, especially if it's a tree crop, can help. But even if there weren't tree crops in there, there could be indicators that kind of give you an idea if there are potential problem areas. Why I like soil maps, and this one is provided by UC Davis, and it kind of overlays on that map. It gives you an idea of what type of soil textures might be in your field. It is not one-to-one. It's not necessarily the best representation of what is happening at depth, but it gives you an idea of the types of soil textures that might be there. Because if they outline and overlay with a potential issue, that can be a consideration. And there are management techniques that can be done if you're dealing with soil textures, which I'll touch upon. Very helpful tools. The first one I'm going to be covering is aggressive pathogens. There's way too many for me to potentially cover, but I will cover a few, at least ones that I've seen. And I'm going to be pulling from examples because I tend to get called out when there's problems, and there could have been potentially something done to kind of identify those problems before they became a huge issue. So I'm not going to cover every single one. But UCIPM is a nice resource if you're trying to get a sense of what could be happening in an almond field. So if in replant situations, it's always very helpful to work with the trees that are there if you have that option. So if it's slightly poorer performing tree, potentially trying to help identify what was causing that poor performance, even though it's at the end of its life history, can help you identify potential issues that might persist into the next orchard. So if you have a declining tree and you shave back a little bit of bark and it's red, it's probably Phytophthora. You have some white matting, oat root fungus. And again, these ones may or may not be able to do something about it. Phytophthora is more of an irrigation control measure. Armillaria root rot, for that, generally speaking, there are mostly rootstocks, because fumigation has kind of been inconclusive from the stuff I've been looking at. You have some flagging scaffolds, and you cut it back, and it looks like there's some black in the middle, probably verticillium wilt, which can be done. But identifying is what's helpful. And to provide an example of those tools that I mentioned prior, Google Earth. This is an orchard I visited recently through the years. I just had Google Earth and I just scrolled back and forth just trying to look at what was happening. So anything pop out to you? What's going on there? I know it looks like a turkey leg, but you can see that both in the prior orchard, in the above photos, there's just this splotch where it just seems like there's a lot of tree loss, which seems to have also maybe kind of persisted into that next orchard, right? So back in 2016, because this is a family plot, they own this land, you could have taken time to kind of look at those trees to see what was potentially causing that issue. Now, I tend to go to the soil map to think there's a soil texture change, but it was basically the same. So potentially you can rule that out. And before I get into any of this, field diagnostics and Google Earth and soil maps is not a replacement for lab tests, especially if you're dealing with diseases. But it can help give you an idea of what to kind of sample and look at. So I went into this orchard later, but this could have been done back in 2016, 2015, and took a look at a tree. So I picked a declining one, just started to take it apart because I was going to be taking it out to begin with. There's some discoloration in the middle, which can be a lot of different things. I know people like to talk about patterns, which, yeah, there's some correlation, but it's never really one-to-one all the time. Something mid-canopy, couldn't really see anything higher up. Seems to be going through scaffolds and into the main trunk area. Notice some gumming at the bottom, so decided to kind of shave it back. So again, if you're not trained in field diagnostics, you can't really do this, but there are farm advisors in the area that can help you with this, and some consultants and PCAs. So there's discoloration that went through about mid-canopy, and then we have discoloration on the exterior base of the trunk with some white matting. Any theories? That's what I assumed, but I took samples and sent them in to be tested, and it basically confirmed that. Now, does a pest mean that that's the root cause? No. But it might give an indication of something. Like Phytophthora isn't just a disease that happens. It typically is associated with irrigation, right? Malaria, though, for the most part, in my opinion, it is kind of more of a disease issue that is there and fairly aggressive and can impact at least almond trees and some other tree crops. So this, combined with the lab test, combined with the shape of the decline, helps me give an identification, an idea about if I'm planting into a crop that is susceptible to this, that it might be something I want to consider. The nice thing is this family, that's just kind of its own block, that the image you see in the photo. So they're able to have a different management zone. So they could potentially do something a little bit different versus having this be a part of 50 acres of almonds, right? But they have that flexibility. And there's a lot of knobs that you can do to kind of help with certain diseases. However, fumigation has kind of been fairly inconclusive, although there's some newer research that might help with that. And generally speaking, prune hybrid rootstocks are less susceptible. I won't say they're resistant, maybe a little bit tolerant. And some of them haven't really been fully tested. And Roger up in Stanislaus, I think, had a study that was looking at some newer rootstocks as well. And generally speaking, prune was it. But again, nothing's a silver bullet. You know, just because it's tolerant or resistant doesn't mean you're not going to have that issue and you should just ignore it. It's a control measure that you have. This is not really helpful, but since I mentioned the disease, I'd bring it up. There's been some research looking at partial excavation of the lower root zone for fruit crops, and at least in the Pacific Southeast. And there is efficacy, but in almonds, I don't really know if it's really practical or helpful, and there hasn't been a lot of research done. If you're replacing a tree, and this is something that is brought up to the grower, there's someone from Clemson University did this research on peaches, and it found it pretty helpful. So if you're, I wouldn't do this for an entire orchard, but if you're replacing a tree, maybe pick a rootstock that's more tolerant of that armillaria. They do some soil pots, they cut out the bottom, so that way after the first year it's easier to excavate and remove it. However, suckering was an issue, and some prune rootstocks are very bad at suckering. So if you're going to choose a prune one, maybe pick one that's been well tested for oat root rot fungus that maybe doesn't sucker a lot. Again, not that I say you should do this for an entire orchard, but if you're doing a tree replacement, I'm just mentioning it. There are many things that can go into impacting your future orchard. One of those are abiotic factors. So this is like things that are not due to a biological organism, so physical and chemical factors. I tend to focus more on the soil because that's the one thing that's staying there when you're planting a new crop. And this is an example of how you can use Google Earth with that soil web. So the yellow lines, the polygons, dictate different soil textures in your field. Again, it's not one-to-one. Never think that it lines out perfectly, okay? And from this image, you can kind of see how in this tree orchard, there's certain areas that are more organically shaped. They don't run down the tree row, where there seems to be less thrifty trees. And generally speaking, they seem to align with that soil web map, which means those textures might be playing a component in those potential trees' less thriftiness. Now, is it a singular issue? Probably not, but it's probably a factor. So what can you do? In this case, the grower decided to split it into different management blocks. You can't do this after planting, but you can do it before if you plan for it. So they took into consideration and they did the field preparation slightly differently, and they have the option to handle the irrigation differently, because that's a control measure after the fact. When you're dealing with different textures, you irrigate to the weakest part of your field, but you have that flexibility and you have that option versus trying to do it after the fact, because it's very hard to retrofit an irrigation system. And again, soil web maps are not one-to-one and they're not great at depth. So in this example, you can see that there are more thrifty trees in certain parts of the orders and less thrifty trees in others. Again, my head tends to go to soil first, though it's not always the main cause. And when you look at the soil map, this area has basically the same soil texture. However, if you dig a little deeper, and I do mean that literally, so I augured down to 4 feet at the yellow and red star, and then the jars that are going to be popping up denote the type of soil that were at those depths. So at 1 foot, there wasn't much of a difference. At 2 feet, there wasn't much of a difference. At 3 feet, what the hell is that? And then when I get down to 4, I start hitting this hard pan. So again, it's all the same soil, but the hard pan is at different depths, because this is nature. Things don't always run in a straight line. And again, I suspected that this was a component to why these trees were generally less thrifty. It's not the main cause. But if they were going to replant into this orchard, it was an identified issue. And I'm not saying you should auger this, but you could probably backhoe it to get an idea to see if you can deep rip it, which is very expensive. But it justifies that management cost. Now, is it worth it to do in this case? I can't really say. That's an individual choice. But you identified it and you considered it as you're preparing this into this orchard. pH is a very uncommon thing, but I just want to show this video because it was a thing I had to deal with. So I'm using these gardening pH tests. Please don't use them, but they're a nice visual for the video. And basically, I tested the soil right where you kind of see me. And the color in the clear window denotes the generalized pH. And again, don't do this. Send it into a lab. I did across the field and at different points. But I went 1, 2, 3, 4, 5, 6, 8 trees down. And that was the difference. So you're at like 7.5, way down there in the video, and at like 5, 5.5, which is pretty comparable. So what the hell would cause that big of a difference 8 trees away from each other? So the high pH is where that red box is, which is what that lagoon was, right? But I decided to plant into it and deep rip it and move it around, right? So corral spots, they're a very small component of it, but you just need to be aware of it. And the thing is, you can move these issues, because when I come out, you have infiltration issues, you have odd leaf systems that are kind of variable nutrient deficiencies, but not because the nutrients aren't there. And as you go down further, those trees are just basically gone. I think the grower is trying to see if they can manage it to kind of bring those trees back. Now, is it worth it? You're talking about 20 trees in a 30-acre block. I don't know. But the problem is, if you're deep ripping it and moving these soil conditions, you could potentially cause little pockets somewhere else in the field. So maybe it's just worth it to avoid. But you identified the issue. And generally when people are doing it, they're doing composite sampling. So they're taking multiple spots, hopefully you're doing multiple spots, and combining them into your field, into a sample, and sending it into a lab. But just by pulling up a map, you can identify problem areas and see if it's worth the extra money just to sample it separately, so you have the information to see if it's worth managing differently. And the nice thing about this block is, like, I didn't want to show all of it because they wanted their privacy, but it had a lot of different land use before the tree crops, and there weren't tree crops before this. So they had this lagoon, they had corral spots, they had agronomic crops, and generally or just piles where they managed the weeds for multiple decades. And that's generally kind of where you saw these weird tree symptoms, right? And again, I'm not saying pH is the main cause, but it was an identification that helped me out. And ideally, you can choose to dig in a little deeper to see if it's worth it, right? Again, because you're only talking about, in this case, a few trees. But I can't really say if it's worth it or not. So again, identification is helpful. But you should always dig a little bit deeper to make sure, because it's a large investment. Because ultimately, the management, especially pre-plant, can be very expensive or very complicated. So you have to really make sure if it's worth it. but you need to identify potential problems to see how big of a problem it could be. So, a large portion of this presentation is dedicated to nematodes, because that's where a lot of my prior work as an SRA was based on. But it is a major issue for tree crops. So, for those of you who don't know, there are various forms of nematodes, both plant-parasitic and not. So I'm focusing on the plant-parasitic ones, because there are plenty of other nematodes out there in the world. They vary in size, life history, and how they could potentially impact the tree. So this is a photo of, I think, a grass root, a visualization with different types of nematodes. Some can burrow into the roots and live in there at different life stages. Some just kind of hit it really hard. But basically, there's different types of nematodes, which I think you all can know that, but just doing it regardless. Sampling is very helpful. So you could use Google Earth, and I've talked about spotty trees and blah, 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 but it's really worth sampling, and regardless, it's probably good to do it. So a good sampling method is to do it in fall, because that's when nematode populations are the highest. And the thing with nematodes is they're not consistently distributed across the field. You have pockets of it, for whatever reason. So like most of you might be favoring the door because you want to leave as quickly as possible, and you're not evenly distributed across this classroom, right? So nematodes are very much the same. So don't think that, oh, that's an equal concentration. I could just take a few samples here and there. So fall's an ideal time because that's when the populations are highest, so you have your higher likelihood of hitting it. You want to get into the soil because that's where they are, so a probe or an auger can be helpful. Probes are easier to clean. In talking to Andreas, the nematologist, he recommends a 4-inch to 24-inch depth for tree crops. And the reason you don't want to sample the top 4 inches is because you're going to want to avoid whatever happens to be growing there that has a smaller root zone. And you would rather focus on where the tree roots are primarily going to be, but you don't want to go too deep. Multiple spots, as I said, nematode populations aren't consistent. I prefer at least 5, and you want to spread it out. So in this case, there are different textures, so you want to make sure you're also sampling within those textures. Do you need to analyze them separately? I don't necessarily think so with nematodes, but this is a composite sample. So you're sampling 5 or 6 locations, you're combining them, mixing them well together, then you're sending a composite sample into the lab, because it's supposed to be a representation of that entire orchard. Sanitize, not necessarily in between each spot, but if you're hopping to different orchards, again, this is happening at one time of the year, so the people doing this might be going to multiple orchards. Nematodes can reside on the sampling things and try and keep it clean, so I tend to prefer just like a 10% or like 1 to 9 chlorine mix, and then rinse it off as well, because you don't want to be moving the problem. If you're sampling an orchard with high level of nematodes, you don't want to be sampling another one and potentially moving, quote, those nematodes into that other sample. So not necessarily within the orchard, but at least within different sampling zones or orchards, it's very important to sanitize. Mark it properly. I can't tell you how many times people have accidentally swapped a number, 6 for a 0, and they switched up the fields. And because at the end of the day, you're getting an Excel table with those numbers that denote whatever your orchard is. So sample properly. And then try to keep it cool. Ideally, you want to maintain them, get them to the lab as quickly as possible, or put them in a thing that can help maintain soil temperature, which is about 60 degrees Fahrenheit. So again, spread it out. What I don't want to see you do is drive your truck along a road, pop in, grab a quick sample, maybe drive a little bit further, pop in, grab a quick sample, and drive off. Those 2 points are not representative of your field. You want a good representation, so get in there and get multiple points, and then combine them. So you send it in the lab, hopefully you sampled correctly, and you'll probably get something like this. The site name's just blocked out for privacy. So generally you'll get multiple columns denoting different plant-parasitic nematodes, some of which may or may not be an issue for your specific orchard, as well as something called free living or whatever. So the one at the end typically is just other nematodes, but it's the other ones that are in the other columns that are a little bit more beneficial. And if you're getting, this is a representation of one sample, and this would be a representation of multiple. So I'm not going to go through every single one of these rows and tell you what I think you should do based on the nematode numbers. I'm just going to go over this one, which generally has what you could maybe consider a low nematode population, plant parasitic, and this one, what you can consider high. But when it comes to nematodes, the ones you're dealing with with almonds. Some need certain management at certain levels compared to others. So this is a generalized recommendation that David, my predecessor, provided, and I haven't really seen too much to counteract it other than maybe the cover crop stuff. But you'll notice that for root knot, ring, and lesion, there are different recommendations based on the number of nematodes per liter of soil. So this is in liters of soil. If your units are not in liters, make sure you convert it if you want to use this table. So in this instance, there's only, quote unquote, only 40 ring nematode, which couldn't, and any level of ring, at least according to David, necessitated some level of fumigation, but at a minimum row strip fumigation, if you can do it. As well as picking a tolerant rootstock. So for ring, you typically want to avoid the more vigorous ones like peach almond hybrids. The P/A counts for peach almond. In this instance, and this is a nice example of the fact that we live in the real world and sometimes there isn't a silver bullet, you have a high level of ring and a pretty high level of lesion. So for the ring and the lesion, that's fine. Fumigation, broadcast, good enough. But when you want to try and pick a rootstock, that's where the confounding factor happens. Because due to the ring level, you want to avoid peach almond hybrids. But for the lesion, peach almond hybrids are actually pretty helpful. So what do you do? So there is no silver bullet, and there's different stages in this orchard's history that have different points for mitigation and management. Fumigation is a pre-plant option. Rootstocks are a post-plant option. You have to consider how effective what you're choosing is at it and manage for the weaker components of each of those with the understanding that you're never always going to get it 100% right. But now you know, so that way you can make informed choices. All right, so again, fumigation is a big thing to consider when you're planting or pre-planting an almond orchard, so I'm going to cover that. So the 4 questions people typically ask is, should I fumigate? I just went over a table that kind of covered some metrics as it pertains to nematodes. What should I use to fumigate? So David did some research with these different products. So even though there are multiple products, what you're really talking about are 3 different active ingredients. So Telon is 1,3-D, chloropicrin is chloropicrin. I think Dominus is some combination of that. I don't really think Dominus may not be really a viable option in almonds. Generally speaking, when you're talking about ring nematodes around here, because this is where I work, 1,3-D and some version of chloropicrin outperformed not fumigating or the abiotic, just generally speaking. I'm not going to run you through the 5 years of data over multiple sites where we came to this overall conclusion. However, there is some considerations when it comes to like applying that fumigant. I mentioned broadcast, so you do the whole field, 100% of it, plant your trees, you row strip it, so you just strip it within the rows, make sure you line it up right. Plant your trees, and then David in the past looked into spot fumigation, but I think that's becoming a less viable option. But basically what spot is you take a giant auger, you auger it into the ground, and then you inject it with the fumigant. It's just right where every single tree is. So the reason this is a consideration is because it cuts down on the cost, because the amount of product you're applying is lower per area. So broadcast you're applying 100%, row strip is about 50, and spot is about 20. But product amount is not just the only consideration. Labor is another one. Spot takes a lot of time. There's more labor associated with it. But the other consideration is how effective each of these treatments are. So David took a look at this, so broadcast, row strip, and spot, and generally any fumigation reduced low disease symptoms. Again, this is ring nematode because this is the primary area for ring nematode. Broadcast only really seemed to help with pin. But in talking to Andreas, pin isn't really a huge concern when it comes to almonds, so should it really matter? From other nematodes to nematodes are not much of a difference. And then generally speaking, spot and row strip when it pertained to yield were about the same. But as I said, the amount of product isn't the only consideration, there's also labor cost as well. Tarps, it's more common now, but back in the day it was less common, so we looked into it. So we played around with different depths, both with PE and TIF tarp. He found, and I think most people know this now, that TIF tarps are the ones that actually reduce the overall emissions. PE tarps don't. They are porous. Plastics are porous. They can let gas through. So polyethylene, that's what PE tarps are, allow gas to go through. So it's not really that helpful in keeping the gas in the soil. Okay? And he did find that certain combinations of depth and tarping kind of have kind of different levels of effectiveness when it comes to tree crops, but I won't get into the overall specifics right now. So another mitigation measure as you're planting into your orchard is rootstocks. So I mentioned as it pertains to nematodes, but rootstocks have various characteristics. And again, none is a silver bullet. But as it pertains to rootstocks, the question sometimes comes up, is that enough to control for plant-parasitic nematodes? So David did a study. So he had a few different rootstocks. He put them in fumigated and non-fumigated plots. And generally speaking, rootstocks aren't as good as fumigation. Again, you don't have to do one or the other. You could potentially do both. So rootstocks in non-fumigated soil had just no fumigation were much lower compared to their fumigated counterparts, right? And this is with ring nematode and a little bit of lesion. So for the non-fumigated trees, the issue is that progressively they got worse. And the thing you need to understand with fumigation and tree crops is that it's just buying you a little bit of time for the trees to get established, because nematodes don't go away forever, they do come back. So by fumigating, you're giving those young trees time to grow larger, so that way they can be a little bit less susceptible. But if you can pick one that's also tolerant or resistant to those nematode pests, that helps in the longer term. And if you're curious about these ones in general, root pack R had the highest disease symptoms, both. It was kind of generally the worst performing one. So again, fumigation is the best solution in the short term, but after 5 years, its effects are significantly reduced, so what you're left with is other management, either post-plant chemical controls or the rootstocks and how well they interact with that. So as I mentioned, rootstocks have various characteristics, okay? So you can consider them horticultural, how vigorously they grow, how well they perform under abiotic factors like salts, how susceptible they might be to diseases, and again, I pulled plant-parasitic nematodes out separately. So I'm not going to go over every single one of these, but I am going to give you an example of an older orchard, because you really have to think about how the shortcomings and the potential pros to your field might interact with the rootstocks and other factors as well over the long term. So David established a rootstock trial in Winton, California on sandy soils that had nematode pressure. It was planted back in 2011 and I revisited it back in 2023. So it's about 12 years old, so middle to end of its overall life. He did a lot of rootstocks, 13 on the nonpareil and then just 6 on the Fritz and Monterey. So I walked this 30-acre plot, every single tree row, and nearly every single tree, and documented just simply, and these are visual observations just to give a little bit of insight, how many trees were replanted based off the rootstock and scion, and how many of them were diseased. And by diseased, I mean they looked like this. It wasn't like a little bit diseased, they were like heavily diseased, okay? So the higher the percentage means there's more replants and more diseased trees. And for the nonpareil, those rootstocks at the top of the list tend to have more of them. And when trying to look at and tease out why, if anything, this might line out this way, generally speaking, there is a high level of vigor with a higher instances of replanted trees and diseased trees, other than Atlas and Catamin. And the higher vigor trees seem to hold consistent as you move across the different varieties as well. So I suspect, again, this is visual data that I'm showing you. It's just insights into how things can interact. But this site was groundwater and it had a lot of nitrogen in the water. So much so that the grower barely had to put any fertilizer on, right? Great. But he had zero control on his nitrogen, basically. How much he could apply and when he could apply it. So this, combined with higher vigor trees, probably resulted in larger and bigger trees, which is not necessarily a bad thing. But the main question remained why Atlas and Catamine were so high. And the thing about Atlas, Catamine, and Empyrean is that they're both fairly susceptible to root lesion, which at the time, towards the end of his research trial, I did not sample in 2023, the root lesion were pretty high, okay? But that didn't necessarily hold true for every single variety, so I can't really say, okay? And just because I'm saying this doesn't mean it actually is. I'm just kind of like walking through considerations and thought processes about how these things might be interacting with each other to hopefully inform a choice you all are going to make that will dictate how things go for the next 20 to 25 years. So one other thing I want to point out. Why is Fritz so low? So Fritz and Monterey had the same exact rootstocks, and nearly all the Fritz had lower instances of replants and diseased trees. And also it was fairly low when you compare them against the nonpareil. What's something about Fritz, for any of you almond growers, that's a little bit different when you compare it to nonpareil and Monterey? I won't grade you. If I put that up, does it help? So Fritz, generally speaking, is more of an upright canopy. Nonpareil and Monterey are more open, okay? So a combination, potentially, again, higher incidences of replants, generally speaking on more vigorous trees, on high levels of nitrogen, typically to be worse on the trees that had a wider canopy. And there are various management things, like when they shake and all that could also play into this. But generally speaking, most likely my hypothesis is that interactions both with the qualities of that rootstock and the shortcomings and the pros within your field can kind of interact into some issue that might be coming down the line. Now I talked to the grower and he was pretty happy with the overall production within the first 5 to 10 years, and yeah, it's dipping off a little bit after that, but it's okay for him. So just because it is an issue doesn't mean it has to be one for you. You just have to make that consideration. So in this case, I personally think that high vigor, high levels of nitrogen, maybe root lesion, all kind of interact together to kind of create this potential issue, combined with potentially also the canopy structure of the varieties you picked. There is no silver bullet. Any other option might have ended up with a worse situation. But you kind of have to think about these things, because especially when it comes to rootstocks, a lot of people tend to think about them singularly. How well do they grow? Are they susceptible to this? How do they do with that? But you have to think about how they might be interacting with other things that may not necessarily be on a chart. But if you want a chart, Roger and Kat and Joe made one. There's some on the back, and it kind of basically summarizes across various levels of research different considerations for rootstocks. So there's a digital version, and I think they have some copies in the back if you want. But as I said, all the considerations aren't necessarily on that chart, and there is no perfect answer. When it comes to a mitigation measure, at least with replant and planting, potentially, if you didn't necessarily sample correctly or capture it, is post-plant chemical applications. So I'm talking about post-plant chemical applications for soil-borne pests, so nematodes. So David, but I basically collected a lot of the data, did a study and trying to see if these post-plant chemical controls were a replacement for fumigation. So yeah, different plots, fumigant with Movento and Vellum, just the post-plant applications itself, and then no fumigation with the post-plant application. So for those who don't know, Movento is a foliar spray. I think it's being advertised as potentially helping with things, while Vellum 1 is injected into the irrigation. And you wouldn't be doing it like in the photos. This is research, so we need localized effects. And generally speaking, the first year overall growth, the fumigants were better. Now with the fumigants with the additional post-plant controls, who doesn't know? But overall significant wasn't clear, but the trees were a little bit bigger with just using the fumigant. And the post-plant chemical controls maybe helped middling. However, when you look at the cumulative yield, and we only looked at this for the first 2 years, the cumulative yield with the post-plant controls were better with nothing at all, okay? Was it good enough? Because this is something you have to apply every year to have it be effective. I can't really say. You'd have to do the math yourself. But these post-chemical controls seem to have an effect, at least when it pertains to yields, if you did nothing at all, but it wasn't necessarily a replacement of fumigation within the first 5 years, because that's when we stopped looking at it. I can't really say year 10, year 12. I also conducted a study looking at what I believe is now called Celebro, and just vellum, no vento, in an older orchard. So I think they were like 7 years old post-planting, or 7 years post-planting. So it was kind of like way after any effects of fumigation and towards the middle of the overall life of the history. Oh, never mind, I put it up there. It was 7 years after, yeah, planting on sandy soils. And the primary pest of concern was ring. So I will note, there's a caveat to this study because I'm using Celebro during bearing. And the label currently is for non-bearing almonds. But again, it gets to its overall effectiveness, and I think vellum you can apply when you're bearing almonds. And generally speaking, there was a lot of data we collected, and for the most part, it wasn't like a clear-cut thing when it came to anything. And the reason 2019 is so low is because it had blast. So in 2020 is when 3 years after we started applying these things every year, we started to maybe see an effect on overall yield for vellum compared to doing nothing at all. This is for ring nematode, okay? And the Celebro maybe if you squint, but not statistically. So for the studies I'm aware of, and my word isn't law, Movento and vellum on younger trees seem to have some efficacy with no fumigation, but it wasn't necessarily a replacement for it. Just vellum on older trees also had some efficacy after 3 years, maybe a little bit. I don't know if it's worth it. And Celebro had weak efficacy on ring nematode. And there's some studies I'm aware of with Celebro has also continued to show that it's generally weak for ring. But for other studies, it is fairly good if you're dealing with root lesion and root knot. But what you're dealing with matters. So I've been covering a lot of pre-plant controls, so I'll mention a few. There is some starve and switch method, so basically you rip out as much of the wood material as you can, which can be an effective strategy for oat root rot fungus, even though you might be moving around. Starve and switch, so you basically plant something that kind of switches what the pest can potentially feed on. These are more towards nematodes, as well as anaerobic soil disinfestation. But again, with a lot of these studies, it might potentially cause other factors that you need to consider. I think with ASD, Phoebe, you were looking at potassium.
It was phosphorus.
Phosphorus, sorry. So again, it may not cause potential other things you need to consider, even though you're trying to manage for one other thing. But at the end of the day, identify what you could be dealing with and their interactions dictates the type of mitigation measures you can take, because sometimes something you wish you did in the past could have helped out. And we can never do it perfectly, but taking the effort to identify potential issues can potentially help with overall mitigation. A couple plugs before I get to questions if you have any. These are my contacts. If you don't have them, cell is more helpful. If you're looking for material, San Joaquin Valley Tree and Vines or SJVTNV is a nice website to go to if you're looking for resources from various advisors from Cooperative Extension. If you didn't know, we have a new production olive manual that just came out. I try to promote these things on Growing the Valley. Phoebe and Luke have a podcast. I've been using the title to help promote these resources if you prefer to go follow more on social media to be updated on these things. I have this fun survey on documenting and mapping out how people say the word almond. It is not a research trial, it's just for funsies. So if you want to add your pronunciation to the map, I have some flyers in the back to answer the survey. But if you look, some regional breakouts. Sac Valley seems to like almond, which maybe many of you already knew, but you can stroll around and take a look. The website is ucanr.edu/sayalmond. I covered a lot of different research trials and nearly a decade's worth of work from my old boss and friend. A lot of different funding sources. I can't possibly even come close to listing them all. But thanks to all of them regardless. And with that, I'm happy to take questions.